Thank you, Tom. So we're going to talk about facts. We're doing subgroups in facts or enrichment trials or basket trials. These all have they're different names, but referring to a very similar kind of concept. As Tom said, I'm Kurt Veely. I'm on a lot of social media. I'll answer questions on social media. So anyway, to dive right in, I'm going to do what I did last week, which was spend a little bit on slides to introduce the topic and then dive right into facts and spend most of the time talking about how to implement this and how to interpret the results. So we're going to talk about a basket trial. This is, we're going to investigate a therapy in 4 rare tumors. So this is a pretty common setting. We have some kind of, often a drug that targets a particular biomarker, and that particular biomarker is present in multiple kinds of cancers throughout the body. And so the idea is that you would enroll a trial in several of these different cancers and see if it works basically across the board. The hope, of course, is since you're targeting the biomarker, that any cancer that expresses the biomarker should be helped by the therapy. But of course, there's always exceptions based on, all the complex stuff that goes on in our bodies. We can enroll here. We've got a budget for 100 patients. We've got time for 100 patients. They're rare tumors. It's going to take a little while to enroll. And we're going to enroll all comers here. So we're going to basically open enrollment to our 4 tumor types, and whoever comes in the door, we're going to go ahead and take them. Certainly, we could do other complex things like try to cap enrollment in certain groups. But for the moment, I'm trying to be simple. Our expectation is that the 4 groups will be about equal enrollment. In practice, you could think about, maybe one of the groups is much bigger than the others, and you may have to work around that as well. We'll hit on that as we go. We're going to randomize 1-to-1 within these groups. This is definitely going to give us small sample size. So 4 tumors, 100 patients, 25, 1-to-1 randomization. We're talking 12 per arm. So we're going to need some big gains here in order to see an effect. And the hope is that, we've got a great new therapy that can do this. So we're attempting to see gains that take us from about 20% response rates on the control arm to 60% on treatment. So we're hoping to really help some people. And at the end of the trial, what we need to do is we're going to identify which groups are promising for future investigation. So we're going to make 4 decisions. So that's 1 decision per group. So the first group, yes or no. The second group, yes or no. The third group, yes or no. Fourth group, yes or no. And of course, this could be future investigation. It could simply be this is to approve in those groups as well. So either one of those puts you in similar kinds of decision space, yes or no on each group. We're going to compare several different designs today. We're going to talk about doing separate analyses in each group. Let's let each group stand by itself. Let's see how it does. We're going to do a pooled or a stratified analysis where this is going to assume we have an equal treatment effect in every one of the groups. So it would allow the control rate to differ in each group, but the same log odds treatment effect. So it's making an assumption that the groups that they either all work or they all don't, which is a pretty strong assumption. We're going to consider doing adaptations or not. So we're going to adapt here by allowing groups to stop for futility or success at particular interims. We're going to keep the 100 here, but we're going to allow certain groups to potentially go away as we go. And then we're also going to talk about borrowing information or not. So the sharing of information across groups, a particular statistical model. One thing to keep in mind here, this is a really complex topic. And the reason is the comparisons between designs are pretty hard to make. None of these designs are going to dominate each other. There's not going to be a situation where, oh, you can always get rid of the pooled analysis. It's never the right thing to do. The separate analyses, it is sometimes the right thing to do. Pooled is sometimes the right thing to do. Borrowing is sometimes the right thing to do. All of this depends on two different factors. And one of those factors is what scenarios do you think are most likely to occur? And by how much do you think they're most likely to occur? Do you think that you're going to get largely different effects? It may work great in two of the groups and not work at all in the other two. Is that what you think is likely to occur? Do you think it's more likely that it might work in all four or not work in all four? Basically, you get these common effects. Maybe that's motivated. You've got a common mechanism of action you're hoping for. So maybe you think that translates into some basically implied correlation between the groups. If I can get it to work somewhere, odds are I can get it to work everywhere. This happens in a lot of antibiotics. If it works in UTIs and skin and abdominal infections, lung infections, if you can get it to the bug, it can kill the bug. So this may be a similar kind of setting here. But what scenarios you think are most likely that, that's not a guarantee. That could go either way. And of course, it depends on how likely that is. If you think it's 98% likely it's all or none, that's a lot stronger than thinking it's 60% likely it's all or none. The other thing that comes into play here is your utilities regarding type 1 and type 2 errors. So, do you think it's more valuable that you want to make sure that you grab all of the groups where it works? Do you really want to avoid the groups where it doesn't? How you weigh those is going to make a difference as well. So this is just a hard problem. In order to try to inform that, we're going to consider scenarios where different numbers of groups work. So either all four groups, the drug works in all four, works in three of them, two of them, one of them, or none of them. And I think that finishes my slide deck. So let's dive into facts. And let me share my fact screen. There we go. All right, I've got some of these pre-populated in case I do anything wrong under the massive public pressure that I'm under right now. But I'm going to go ahead and try to do all these from scratch. All right, so Facts 8, new window. For the screen, we're in enrichment and basket trials. And you can see we've got continuous dichotomous time to event as we've had in prior versions. Remember the new thing that's in Facts 8 that's really cool is all of these templates. So if you want something that's pre-populated with different kinds of design, so basically it's already filled in a lot of the boxes for you. You can now pick one of these templates, so it'll open up and you can immediately see it. I'm going to try to do this completely from scratch here and go through basically an overview of what all the boxes are doing. But there are these quick starts that allow you to get going. And so, we could do this basket trial with 5 groups in borrowing, and then we'd have to make some more minor tweaks, delete one of the groups to get to 4 groups and so on. We could do it either way here. I'm going to build it up from scratch. So new dichotomous, here we go. All right, so usual fact screen. I've got to say lots of stuff about my study. So I'm including a control arm. This is a randomized study. If you had a single arm trial in each group, you could compare to a number where I need to show that each group, whether it's greater than 20% or not, and just fixed. But this is a randomized trial. Responses are good. Certainly if it were deaths, you would say responses are bad. Superiority trial, we want to show we're better. I'm not going to do adaptive features. I'm not going to do longitudinal modeling, but that is all available to you, as are some other bells and whistles, such as these kind of Markov models, which essentially allow you to have a longitudinal structure where patients, for example, if they have an event, they may stay in that category. But this Markov model is a particular kind of longitudinal model in here. The big thing you have to fill in is the groups. You got to tell it what groups you're interested in. I've got 4 of them. And you'll notice that each of these groups, there is a potential cap on each of the groups, and there's a potential cap on the overall study. So when these are checked, this is saying that you're going to have a limit on how many patients can be in each group. So with all these checked at 50, it says whatever I do, I can't have more than 50 in each group. I could also put a cap on my study. And you got to be careful and make sure that these things agree. Fax will yell at you if you don't. So you can see this is how Fax yells at you, this little red exclamation point. And of course, the reason that it's yelling at me here is I've got a cap of 300 for the overall sample size, but I can only put 50 in 4 groups. So that's only 200. I can't get to the 300. So these aren't particularly, anyway, they're not consistent with each other. I could change that to 100 and my exclamation point goes away. I might do something like this where I didn't want any of the groups to dominate. We've enrolled trials like this where one of the cancers is much more common than the other. And we don't want our trial to enroll, 99 of group 1, and I've got one stray person from one of the other groups. And so, I go back to the FDA or the EMA and I go, hey, I got 99 in group 1. I want a label for everybody. And they're like, you didn't investigate everybody. You investigated group 1, which is a perfectly reasonable thing to say. So anyway, you can put caps on it and say, I don't want any of the groups to have more than 50 patients, but the overall cap is 100 in this case. I'm going to actually just take these off, and I'm going to take whatever actually enrolls. But you can mix and match these. You can put caps on some groups or others. You could put caps only on the group and not cap the overall study size, in which case the trial would try to enroll to the cap in each group. But you got lots of flexibility here. This one's simple. It enrolls everybody who walks into the door until I get 100 patients. All right, group info here. So when we're doing inferences, Fax is going to compute probabilities. So it's going to compute probabilities, say, is the treatment group a certain amount better than the control? And so we can have different rules for futility and for success. I'm going to change all of these to 0 because it's the simplest thing to do here. And so that's just going to say, all we're going to be interested in is the probability that the treatment is greater than the control. So we're doing straight probability of superiority. If I wanted some kind of super superiority on it, I could say, look, I want to make sure that I am 10% better than the control, for example. So I could put that in. We often do that kind of thing for futility, where we might say, hey, it's not really a meaningful effect unless you're 20% better. And so when we're computing futility, we may say, hey, if we don't have a good chance of getting this meaningful effect, we're going to give up. So in this case, I'm just going to leave these at 0 for simplicity, but you have the option. You can also talk about the phase 3 success probabilities in here. So you could say, here's the size of the phase 3 trial that I'm going to run. Here's the alpha. Of course, phase 3 is often 2.5%. And if you need a super superiority margin, you can put that in. You can also put non-inferiority margins as well, as the case may be. I'm just going to consider, straight superiority trials. These are all 0 here. I do have to say I don't have longitudinal modeling, but I need to give it a visit. I'm going to say, I think for this example, I have a 12-week endpoint. So put a 12 in there. Could be 26, could be whatever it is, could be instantaneous. I'm not doing any variance. You can change the sample size and let it experiment with multiple sample sizes within the same file. But again, we're going simple today. Don't have to use all the features and facts for every trial. My virtual subject response. So let me add one. And let's start with it works in everybody. So my control, that's just by luck. I already have the 20% populated. I'm trying to get to 60. So 60 in all of them. Suppose it works in 3. All the groups I'm considering symmetric here, so it doesn't matter which 3 I pick. So, and then we'll do 2. And let me make sure I didn't change that 0.5 to a 0.2 for the one that didn't work. All right, 2 work. 1 works, I guess. 0.6, 0.2, 0.2, 0.2. And finally, our joint null, none work. And this is 0.2 across the board. And of course, I don't have to have, we could say things work to different degrees. Some groups could be 20 versus 60, others could be 20 versus 50. You can mix and match these things as well, as much as you would like. You can bring in an external file. So if you want to design your own kind of data simulation in R or whatever, you can bring that data in, not using that particular feature. My accrual, I'm going to say this is a little bit slow enrolling. So let's say I get 0.75 patients a week. So I need 100 patients. So is that 133 weeks? So that's it's a bit 2 years plus, 2.5 years maybe, in order to get all these patients enrolled. As with all in facts, you can put ramp ups, ramp downs. I'm just going to have this uniform accrual rate over time, but you can basically make this look however you would like. The other thing that you can do here is you can specify that some groups are more likely than others. So if one of these groups was 70% of the population and another one was only 10, you could put those numbers in. And these are all relative. So right now this says 1 to 1 to 1 to 1. They're all equally likely. But this could be, 4 to 3 to 2 to 1, and 40% are in group 1, 30 in group 2, 20 in group 3, 10% in group 4. And you get a little graph here showing what it expects the enrollment to look like over time. You can also delay groups. So you can say, look, I'm going to start enrolling the first 2 groups, and groups 3 and 4 may come online later. So again, this is all intended. In a lot of trials, you wouldn't use all these features, but it's there for the trials where you do need it. No dropouts in this. Certainly the usual with facts. You can do dropouts by arm, by visit, by groups, in all kinds of different combinations. All right, my design. Okay, let's start. The simplest thing for us to do is the separate analyses and the pooled analyses. And so in order to do the separate analyses, I'm going to say, hey, let's model the controls separately. So in this case, I need to, it's dichotomous, so there's a rate on the control arm. This models everything in terms of the log odds. So I need to put a prior on that log odds. And I'm going to put a 0-10 prior, try to be non-informative here. A normal 0-2 looks kind of uniform. on the actual rate. So 0.2 on the log odds looks like approximately uniform on the rate. Really, it's like a normal 1.81 or something is if you want to get as close as possible. But I'm going to do more like a non-informative, really high variance. This looks kind of like a beta with small alpha and beta floating around in here. So hopefully this will drop out relatively quick. And let's see. All right. And then I also want to model the group separately for the treatment arm. So we set a prior for the control arms. We're going to set a prior for the treatment arms as well. I'm also going to do this. And there's also this across group priors. This is where we get our pooled analysis. So facts will always fit this model where it will say this theta across groups, this is the log odds under the assumption that the treatment effect is the same in every single group. So this is going to be, I'll set this to be pretty non-informative as well. I'm not going to do frequentist analysis here, but it's available. Here you can do allocation. I'm just going to do 1-to-1. Usual allocation that we see in facts. The sum of these numbers is the block size in each of the groups. So if we do 1-to-1, this is blocks of size 2. If I do 6-to-6, it's blocks of size 12. So I'm going to go ahead and do really small blocks here, be sure I get equal values. In practice, you'll have to decide what's good for operations here. Certainly, this is good for the statistical aspects. All right, my success criteria. Let's pick something pretty basic here. We're not doing anything adaptive. Those numbers where I filled in the study CSD for success, that's where these came from. So they've shown their head again. I'm going to say that the trial is successful if my probability of superiority is greater than 0.975. And I'm going to do that for each of the individual groups. And I'm also going to do it for that across groups analysis. And let's see, I'm going to do the same thing up top. So if a group or the across groups is not successful, it's going to be viewed as futile. You could put a gray area in between where something successful, really low is futile, and in between is some kind of inconclusive. The other thing that you can do here, there's lots of different options on the screen. You could, these check boxes here where it says a study is a success if all selected groups are a success. It may be the situation in a basket trial. Suppose one of the groups is bigger than the others. Suppose you really need that group to win, or you're not going to have a marketable drug. So the market will simply not be big enough. We won't be able to run a phase 3 unless we can get group 1. Then we might want to say, hey, the study is successful if I can get group 1 in this case. So that is the kind of things that you can check here, and you can also check it for futility as well. Here, I'm just going to get the straight group decisions individually. But there are lots of ways to combine things. All right, let's simulate this. I'm not going to do the biggest sins. Let's hope nobody is on our grid to stop me from doing this. Usually the grid lets us do lots of things at once. Oops, okay, so call this temp. Yep, I have lots of temps. And there we go. Okay. So we'll give this a second. Remember, what we're looking for here is the probability of getting a win in each of the individual groups. We're making 4 decisions. So what do we got here? Come on, 3. You can do it. All right. Let's see. What do we got? Okay. All right. So let's see in each of these scenarios how this is done. So you can see all, none, and then 1, 3, 2. They're in alphabetical order here. Okay. So I've got in these 4 columns the probability that I declared success in each of the groups, and this is from these separate analyses. So I'm making only looking at group 1's data to analyze group 1, only group 2's data to analyze group 2, and so on. And you can see we've got about 60% power in each of the groups. They all have the same VSR, so they're all identical. The differences here, the 0.594, the 0.626, those are purely simulation error. If I ran bigger sims, these would get closer together to whatever value around 60% is the truth. You can see in the none work, I've got about 5 to 6% type 1 error. I put in the 97.5% for success. This is a discrete setting. So of course you often don't get 0.025 exactly. This is actually up a little bit. So that may be acceptable to me for a phase 2-ish kind of trial. 5 to 6%. If I needed it to be lower, I might need to change my threshold in order to get this. The other reason I'm happy with this right now is I'm about to show you effects of the other designs on type 1 error. So having a little bit of room to show what moves is valuable to me. All right. And then, of course, since these are all separate analyses, these 60% power, 5 to 6% type 1 error, they apply across the board. So in, say, the 2 work scenario, I get the 60% power for the 2 groups that work. I get the 6% type 1 error for the 2 groups that don't. So 60% power isn't that exciting. We'll see if we can do a little bit better than that. What about the pooled analysis? All right, that's this probability of success across groups. All right, so remember, this is where it pools the 4 groups together. So instead of having about 12 observations or 12 in each arm, this pulls everything together. We get about 50-50 from our 100 observations. This power is great, 99% when they all work. So that's, hey, that pooled analysis is wonderful. Type 1 error when none of them work, that's about 3%, 3 to 4%. That's not bad. All right. So then what about these other ones? All right. So when 3 work, 92%. Okay, that's high power. But what does that mean? So it's a pooled analysis. So when 92.4% power, what that says is there's only 2 kinds of decisions you're going to make. You either say they all work, happens 92% of the time, or you say none of them work, which happens 7.6% of the time with simulation error attached. So that's a pretty big type 1 error for that group that doesn't work. So the chance that we're going to carry this group 4 forward, or say we want it approved, even though it doesn't work, is 92.4%. Similarly, in, say, the 1 work scenario, our power is 23%. So that says, again, 23% chance we're going to carry all of them forward. We've made 3 type 1 errors. And then a 77% chance we take none of them forward. We've missed that 1 group that works. So all right, so let's see. Okay, so I think we've got that. All right, so 60 and 5, let's keep those numbers in our head on what we might want to do better at. So let's try to add borrowing to this. And remember, the nice thing about FACS is getting the fixed trial is a large amount of the work. We've seen how to get most of the information in. We've seen the kind of operating characteristics we're going to care about. So now the question is, what do I click on to get the borrowing? And what I'm going to do, if you want borrowing, so a hierarchical model, remember the idea is that we know when we're dealing with multiple groups, there's an advantage to doing shrinkage estimation. Whenever you're looking at results from multiple groups, the highest value is usually biased high. The lowest value is usually biased low. It makes sense to pull those groups together in terms of the point estimates, and you can get better estimation. This goes back to Stein estimation in the 1950s. And so we're going to try to do that. To use the hierarchical model across groups, you click hierarchical model across groups. So let's change this. We're going to need to do some more sims. I've got everything pre-cooked in other files, so it's okay if I delete those. The hierarchical priors here, these require some experience in order to set. When you're dealing with a dichotomous value, the center of the hierarchical model doesn't matter all that much. So I'm going to give it kind of a non-informative prior. But the tau parameter, this is the across group variance. So part of my prior is I need to say how different do I think these groups might be. If that across group variance is tiny, that would say that I expect there to be very, very, very small differences between the groups. So in the extreme, a tau of 0 is basically heading me towards this across group prior. I'm assuming that all the groups have the same effect or tiny, tiny differences. On the other hand, if I pick tau to be large, what I'm saying is that there's a large across group variance. And so that might get us closer to the separate analyses. If I expect the groups to be quite different, I may not do much of the shrinkage. So, of course, the issue always is what should I set that prior to be? And in practice, what you want to do is think about what your goals are. What scenarios do you think are most likely? How do you value these type 1 and type 2 errors? And we often set priors in order to get good operating characteristics. Of course, if you have an expert who knows, how different these groups are, you could set it that way as well. But we often do let operating characteristics drive our decision making. All right. So I'm going to pick priors 0.3 for the center of this and 0.5 for the weight. If you've seen other talks that we've given at Berry, this is a pretty common default prior for dichotomous settings. It works pretty well in a lot of places. So I'm going to go with that. In this case, the 0.3, remember this is on a log odd scale. So it's saying there's some similarities among the groups, but it allows some differences. The weight here is the effective sample size in terms of the number of groups. So I'm basically saying this is equivalent to having half a group of information, and I have 4 groups that are supplying me data. So hopefully this can get moved a little bit in response to the data. All right, that's all we need to change to get the hierarchical model. I'm going to leave the controls being modeled separately. So perhaps the baseline differs among the groups, perhaps not, but I'm going to leave that alone. I'm getting the borrowing from the prior on the treatment effects here. So let's see. Go back to, whoops. Keep everything else, can't click. All right, let's simulate all these again. See what we get. Give me my results. There we go. Okay. All right. So let's see what we got in each of these groups. And remember, these are always nothing's going to dominate anything else here. There's always pros and cons, and you have to decide what's most likely and most valuable. All right, if they all work, so remember we had 60% power before. All right, the borrowing looks pretty good here. These are all almost 90 now. So 60 to 90, that's it. Okay. Okay. Sorry, I had a dinging floating around. I couldn't find where it was. All right, so let's see. So we've got 90% power here as opposed to 60 within each of the individual groups. So huge gain. If none of them work, we actually get reduced type 1 error. So the reason that the borrowing does that is, remember, it's shrinking things together. So the reason you often get type 1 errors is you get this outlying value. You got 4 groups. There's a fair amount of random highs that happen among 4 groups. But if you have 3 other groups that are null, they pull those random highs back. And so you actually eliminate a fair amount of the type 1 errors. So those 5 to 6 rates are now, looks like 1.8 to 2.5%. And again, if we ran more sims, that would converge to something in the 2-ish range, I assume. All right, so that was an advantage. That's good. All right, and then the hard cases, of course, are where you have mixed results. Because when you're pulling things together, you're pulling some things that work together with some things that don't. And so you expect there to be some trade-offs there. In this 2-work scenario, you can see it's 60% power, so that's about the same. We've got some inflated type 1 error here. So the 6% went up to 13.9, 13.6. So those are increased. So we're doing a little bit worse here for the type 1 errors. But of course, keep in mind the type 1 errors are still nowhere near what the across group type 1 errors were. So these were 64% rather than 13. So the hierarchical model has much less type 1 error than the pooled analysis does, for example. And you can see these same kind of trade-offs across the board. So when 3 of them work, we get higher power for it's hard for me to actually there we go. I'll try to do that. We get the 60% power going up to 77, 79, but we also have the type 1 error going up to 26. So you need to decide how you value these things in practice. One, I'm not saying it's the best, but a simple way that you could value them is I just want to calculate how many groups I get correct. Here for the first group, I've got a 17% chance, improved chance of getting the group right. Another 17% chance of getting the group right. Here it's 19%, obviously some simulation error. But in total, I've, added about, the equivalent of an expected 0.55 groups correct in these 3. And I'm compensating for that because I have about a 0.2% or 0.2 probability of getting this group wrong. So if I were to just compute the average number of groups I get correct, in this scenario, the hierarchical model has increased it by 0.3 or so, 0.3 groups. Certainly in the all work, that kind of metric would be even better. This is improved by 0.3, 0.3, 0.3, 0.3. So I'm almost improving it by an entire, 1.15 groups or so. Here, the null I'm doing better, but you can see the trade-offs involved. In one works, for example, my type 1 errors are about equal, but I've lost some power here. So I've made things a little bit worse. And so you're going to have to weigh, do I consider the gains in some scenarios to be worth the losses in others? So you have to weigh, what do you think is important here and what's likely? If I think I have a common mechanism of action, maybe I think the all work and the none work are far more likely than the other possibilities. So I want to value the gains that I'm getting in those scenarios. Maybe I'm worried that things are going to be all over the place, in which case maybe it might worry me. I feel I'm going to be in one of these, the 2 work scenario, and maybe this isn't as valuable to me compared to, say, running separate trials. Another thing that you might consider here, which I'm not doing in this talk, is you could weigh type 1 and type 2 errors differently. So you could say, hey, this 26% type 1 error, that's really, really horrible. And this 30% increased power I don't care about. Or it could be the reverse. I want to make sure I get all the good groups. But one of the things that you need to do is you need to think long and hard, what is the goal of my trial? Because whatever decision you make, you are making an implied utility. Each of these designs has its strengths, and by picking one, you're saying, I value X over Y. And that should be a conscious decision rather than an unconscious one. All right, so we've got borrowing. Let's see if I put adaptations on here. So let's see. So what I'm going to do to adapt is I'm going to run interim analyses, and I'm going to, at each of the interims, I'm going to allow groups to stop for success or futility. So if the results are really good, I can stop at an interim, just that group. If the results are bad, I'll stop for futility, and I'm going to keep my 100 sample size. So I'm going to, any spots that I clear in my trial, I'm going to enroll the other groups to try to get answers for them. Now, that may not be what you want to do. In some cases, you just want to reduce sample size. The reason is that you can imagine if you stopped 3 groups early and then you're only enrolling a 4th group, well, that could make your trial in terms of time longer because you've got to wait to enroll that one group, and that's all you're enrolling anymore. So there are some trade-offs in your enrollment decisions here as well. But at least all of this is easy to do. I need to, on the study tab, turn on adaptive features. What's that? All this is pre-cooked. I'm not worried about those. And then in the design, I need to add interim analyses. I'm going to do these at information, and I'm going to do them at 50, 60, 70, whoops, can't type. 50, 60, 70, 80, 90, and 100. And the reason those exclamation points, if you do things out of order, it gets mad at you. So you have to have the interims going. Interim 6 has to happen after interim 5 and so on. All right, so I've got my interims. And then what I need to do is at the interims, I'm going to allow stopping for both futility and success. I'm going to stop, and I can say I'm only doing this for some of the groups. So I'm going to allow each of the individual groups to stop if I'm above 99% probability of superiority. And you probably want to tinker with this. The more aggressive you are, the faster you stop. Smaller sample sizes, but of course it takes, the more aggressive, so the more aggressive, the easier it is to stop early. Potentially you make more errors when you're more aggressive about doing it. Same for futility. Let's see, I'm going to put 0.3 in here. Remember, these are probabilities of superiority. So if the probability of, if the treatment effects are equal, So I observe the same rate in control and treatment. That's going to be basically, I don't know which one's better. So that would be 50-50. So when I say I'm going to stop if the probability of superiority is 30%, at least in the separate trials, I need evidence of harm in order to actually stop these. So I may want to go bigger, but I'm not going to go bigger here, but you can change these values and experiment with what it does. Final evaluation, I'm keeping the same. And let's run this. It'll take a little bit longer. Oops, simulation. A little bit longer because we've got all the interims in here. Let's see what we've got. So the hope here, at least, is we save some sample size. Maybe it changes the accuracy of the groups a bit. Sometimes it does, sometimes it doesn't. Looks like in the 2 work scenario, these were about 60 and 13 so the power is still about 60. Type 1 error went up a little bit, but not much. In this case, looks like we gained a little bit of power in the 3 work scenario. Type 1 error maybe up a smidge. I think it was 26 before, so 28. Gained a little bit of power. Anyway, it looks like we're gaining power across the board, and maybe we're gaining a little bit of, gaining's the wrong term, we're getting hurt a tiny bit on type 1 error. So basically, it looks like things are a little more likely to declare success. The main thing that's changed here is not really the probability that each group is declared to be successful. Instead, it's the sample size that's being changed here. So in the all work, we're down to an expected sample size of 75, 85 in the null. So we're saving 5 to 25% of our sample size, depending on which scenario we're in. So again, if we're valuing, you can kind of see where the hierarchical model is going to be the right choice. If we think that the all or none are more likely, but these other ones are possible, the hierarchical model is really doing well in these scenarios, and it's not doing that bad in these scenarios than the separate analyses. So, if you do all the integration over what's likely, the hierarchical model may win. Certainly, if you think this, the 2 work is the right scenario, separate's probably the way to go. If you were absolutely sure that they either all work or don't work, then the pooled analysis. We got this 99.6 power. This is great. But of course, it comes with really, really bad decision making if you're not in one of those top 2 scenarios. So high risk, high reward, depending on how sure you are of these scenarios. So again, it's important. You probably need some kind of scoring rubric. How do you value this? That rubric could be complicated. It could say, I'm going to run a phase 3 trial in whatever groups that I say work here. And so I may want, my metric could be, do I win that phase 3 trial after I'm done? I may want to go ahead and do that kind of analysis. And I may pick basically what sets me up best for success in phase 3. One of the possibilities here. As a final trial, I could also consider, can I just do the adaptations but not do the borrowing? And the answer is yes. So all we've got to do is take this hierarchical model off, groups modeled separately, and we're back. This is what we entered before. Fax remembers what was in each of the boxes, even if they go away temporarily. And so we'll simulate this. And so I think this goes back to about the same power and type 1 error values that we had before, but again, we save a little bit of sample size from being adaptive. So while I'm here, I just want to harp over and over and over again. it's worth thinking, what is a win, in order to decide what design you want. Actually, it does look like we gained a bit of power out of these, and maybe a little bit of type 1 error as well. So it looks like from 60, we're up to 70. For power here, type 1 error went from 5 to 6 to, maybe we need to run more sims to check this, but it looks like 6 to 10. So, there's a trade-off here, which I could get from the fixed trial as well. And here you can see we're not really gaining a ton of subjects. So the separate trials, they're all small samples. You don't get any of the benefits of pooling information. It's hard to make early decisions in any of these groups. So the adaptations don't help you as much here in terms of sample size. All right, so I think that's the menu here. Conclusions for the day. Know your goals. You've got lots of options. Borrow or not borrow, adapt or not adapt. Separate trials across groups, all kinds of things that you can do here. So good time for questions, Tom.
Thank you, Kurt. Well, we have a brief answer too, but as we haven't got any others, I will give it to you. In the pooled group, what is being compared? Pooled dose versus control?
No, so it's basically fitting a model where every group has its own control rate, and then it assumes that the log odds difference between treatment and control in each group is the same. Now, I can't do log odds in my head, but it would be, at least on a probability scale, it would be like saying one of the groups is 20% control, 30% treatment. Another group is 40% control, 50% treatment. So those are the same differences, only instead of having the difference be on the probability scale, it's the same difference in the log odds scale.
And is there effectively is the across groups? Are they weighted by the group sizes? If you've got a bigger group, it'll have slightly larger effect on the pooled.
Yes. So that's certainly, it basically is fitting a kind of linear model that you would see if you were doing a logistic regression and you just had basically separate intercepts for each group and a common treatment effect. It's fitting that kind of linear model. And so if one of the groups is much, much larger, you can imagine if one of the groups is 1 million patients and the other groups are 10, it's going to take the 1 million patients, I can estimate the treatment effect off that. I'm assuming they're all equal, so that would definitely dominate the groups there. So,
thank you. And a question from me: do you know? So when you're doing the borrowing across the groups, and you're having the early stopping. So the early stopping of the early stopped groups the results will still be in the estimate of the hierarchical model.
Yeah.
So is there an effect of that early stopping on that borrowing? I suppose you might. Yeah.
Although, of course, they have smaller sample sizes, so they'd have less than the groups that are continuing, but they will have a continued effect on the model.
Okay, that's all I could think of to add into the questions. Thank you very much, Kurt. Very clear as ever. And look forward. And thank everyone for attending. I hope that was interesting and informative. I will attempt to get the emails sent out tomorrow with the links to the recording if you wanted to share. And hopefully we'll see you next week for I think it'd be number 7 in the series, which is, let me just check. phase 2 platform trial. So screening, if you're the setup is in a large pharmaceutical company, you've got, you can see multiple assets coming along in the future, all in the same indication. So maybe you can more efficiently do the phase 2 testing by setting up an internal phase 2 platform trial. And we'll be looking at designing and simulating that and the issues around that kind of designing that kind of trial. So thank you very much. I'd hope to see you next week.
Thank you, Tom.